Hello, my name is Renata Cabello. I'm a bioinformatician in a group of Alfredo Iacquangeli, and my title for today is Transcriptomics Analysis of Postmortem Motor Cortex Reveal Altered Neuropeptide Signaling Processes in ALS Patients. I'm going to start this presentation by showing you this picture from the ALSOD database, which is maintained here at King's College London. This is showing a ever-growing list of definitive ALS genes and how more and more genes are being added to these ALS risk factors genes over the years. And what is interesting is that most of these genes have been discovered here, and if it's not the principal investigator from King's College London, and it's probably one of the collaborators from King's College London. So this department has played a very big role in discovery of these genes. And as I said, I'm not going to talk about these genes today. Despite the fact that we know we identified these many variants linked to ALS, the diversity of processes involved and their relative importance in ALS pathogenesis is not clear. So today I'm going to talk about ALS and genes, but not genes as risk factors, but genes which are being expressed in ALS patients in a different way than they are in controls. So the goal here of this study was to use two large post-mortem brain RNA-seq datasets. I will explain all these terms. Of ALS patients and controls in order to obtain a gene profile of ALS that would identify the underlying molecular footprint of ALS. And of course, all that in order to propose novel hypotheses about disease mechanism and potential drug targets. So I said RNA from motor cortex and promised to explain. So why RNA? Well, in the first slide, when I showed you the genes discovered as risk factors for ALS, there we're talking about the DNA of people. It's static. It's a risk factor that you're born with. Now RNA is giving us something completely different. By sequencing RNA versus DNA, we get a snapshot of gene activity in our sample, which genes are being expressed at the moment of sampling. So it's a very dynamic picture where we are working with RNA-seq. And why motor cortex? Well, motor cortex is affected from the earliest stages of the disease, and it's one of the brain regions involved in the pathology of ALS. So it's a very good region, tissue to study ALS. And luckily for me, I had access to two very large datasets of RNA-seq of ALS patients and controls. So the first one is our in-house dataset, King's College London and MRC, Neurodegenerative Diseases Brain Bank. I believe that the dataset, the smaller dataset, the ALS dataset that I processed from there was processed by Ashley Jones and Claire Trokes here several years ago. And the second dataset is called Target ALS. I've taken a subset of that, and I'll tell you what I took. It is a very large multi-omics resource, publicly available upon request. And it has all sorts of data there, phenotype data and DNA-seq and RNA-seq. It's a very interesting resource, and I was, again, lucky to have access to that. How are these generated? Just very briefly, how are these generated? Tissue has been extracted, RNA has been collected and sequenced. So I'm not going to go through into any details of any of these steps. Once this RNA has been sequenced, it's stored in files to be processed by bioinformaticians like me. So when I say that I've used the brain bank, like no freezer was involved in any at any step of my analysis. I'm looking at the files. Yes, very large files, but only files on a computer. So what did we do with these files? My first data set, this in-house King's College London data set, there were 171 subjects. And the second one had 132 subjects, but I had very close brain regions from these two data sets. So I took multiple samples, and I had 234 samples. We passed these samples as files, through very standard bioinformatics pipeline for RNA-seq data. So without any really technical details, the first part is just to make sure that things are, quality-wise okay, and to simply count how much of each gene there was in every sample that I took. And so this will give me these gene counts per sample, something like this. You see, so I have this, let's say, put a few genes in a few samples. In theory, I would have, tens of thousands of genes and hundreds of samples here, as you saw. So I have a value for every gene, even if it's a 0. I have a value for every gene in every person. And now differential expression part of the analysis is here to tell me what is different between my cases and my controls. So I'm looking at this matrix and I'm trying to decide what is different. And finally, these genes I have a list of genes, but this is a static list of genes. I want to see what do they do that is in common, what pathways, what processes are these genes involved in, what do they do. So this is called either enrichment, or once upon a time they called it pathway analysis. So this is what we did here. Again, we process the data set to get these gene counts and check what is different in cases and controls, and then comes the hard part. What does it mean? What does it do? So this is my methods here. If you now let's look at what we found. Here we see genes differentially expressed between ALS cases and controls. This is volcano plot is like the most typical way to present results of differential expression. So we have on x-axis fold change, which means how much is this gene changed in cases versus controls. So higher fold change means that it's overexpressed in cases. Low fold change means underexpressed, and it's log 2. So if you look at value of 1 means that it's double, then value of -1 means that it's halved in an expression. So I'm interested in fold change, and I'm of course interested in p-value, which tells me whether it's significant. So it's my corrected p-value that is interesting to me. And here what is interesting to me is the red spot. So every spot is one gene. I have here one data set, here the other. Every spot is one gene, and I want to look at these red genes that are passing the filter for both p-value and for fold change. So I want to see what are these doing in one data set, what are these doing in the other data sets, are there any in common? So these are the questions that I ask. So let's start with the numbers of genes that we found. You see, going back. Oh, I can go. Let's start with the numbers of genes that we found. So it's over 1,000 genes in the KCL Brain Bank, and a bit more than 400 in target ALS. 33 genes are in common. It is a statistically very significant overlap between the two genes, considering that we have 30,000 or so genes. And slightly different picture in what is in red is what is upregulated, in blue what is downregulated. So in KCL Brain Bank a bit more upregulated, in target ALS a bit more downregulated. 33 genes that are in common. Was good that each one was it was always the same upregulated or downregulated in two data sets. It never happened that it was upregulated in one, down in the other, which is already good. So I do have some intersection. Now, what are these genes? So usually when I present slides with genes, and I sometimes leave it just for like a minute for people to look at and recognize favorite genes. These in blue are downregulated, in red are upregulated. These that are in bold are the 3 genes that are in bold because just a few days ago I had another data set that is not brain, but it's a blood data set. It's a very small data set, and these 3 were significant in that too. So I'm keeping them in mind as I found them also in blood. So what are they involved in? When we looked at each one of them, it's a small list, so manually we went over the list, and they're involved in neuropeptide signaling, neurovascular unit dysfunction, neuroinflammation, neurogenesis, metabolism, ubiquitin proteasome system, autophagy, etc. So one thing is to look at the genes in isolation, to look at the these 33 genes, but what interests me more is insight into the collective biological function of this underlying list of genes. To do that, you can use many tools, but luckily through King's College London Genomics Center, I had access there, cutting it now. I have access to this Metacore tool that once upon a time I used and I really liked. So for a small pay-as-you-go price, I had access to that and used that for enrichment. So there are various enrichments that this Metacore is providing. So let's start with this one. It's process networks enrichment. It is this is proprietary to Metacore, to now, which is now Clarivate Analytics. These networks have been assembled by them, and so the graph in the top is King's College London, the bottom is Target ALS. And is there something in common? Yes, actually, the only one that is significant in Target ALS is signal transduction neuropeptide signaling pathway, which is also among the top 3 in a brain bank. So that's really encouraging that I have a very significant pathway in common on this in these both data sets, and even more, it's an ALS data set. So the fact that I will find altered neuropeptide signaling pathways is very encouraging to me. Now, apart from this process networks enrichments, I've performed several other enrichments. Here, with all these other enrichments, they're telling me the same story, but 2 data sets are telling me different stories. So, according to all other enrichments, which are diseases, gene ontology processes, reactome pathways, it's King's College London Brain Bank dataset is enriched for everything to do with central nervous system. Even diseases are heredodegenerative diseases, Huntington, central nervous system diseases. Go processes are relevant to the development of nervous system. Reactome pathways related to nervous system functioning, such as transmission of chemical synapses. On the other hand, Target ALS seems to be really a dataset enriched for processes, diseases, pathways related to immune infection, to immune response. Be it diseases, various viral infections, response to bacteria, response to virus, even coronavirus, response to coronavirus. Gene ontology processes related to immune defense, reactome pathways pertaining to interference signaling. So it's these data sets are quite different, and both do make sense, in particular KCL London, because of the nervous system involvement, and Target ALS also immune. I'm singling out one gene here, which is melanocortin receptor 4, MC4R. Why is this gene singled out? There were 33 that were in common. So that's the neuropeptide signaling pathway, which is among my top enriched in both datasets. This is melanocortin receptor 4 is in these 2 pathways, and it happens to be upregulated in both datasets. We know that the activation of MC4R induces hypermetabolism, increased energy expenditure, cachexia, and these are 3 processes associated with increased lower motor neuron weakness and poor prognosis in ALS patients. And we also know that certain blood-brain barrier permeable MC4R antagonists, such as TCM CB07, they have already been shown to prevent the progression of cachexia by arresting the MC4R activation. So here we see that MC4R is clearly a plot of its expression in cases and in controls and cases of both datasets, and it is upregulated in both. So we propose it as a gene to keep an eye out on as maybe future biomarker or in screening studies. As you saw on the last slide, expression plot of MC4R and its expression levels in cases and controls, we are actually providing access to our entire result set to the wider community through this ALS Gene Expression Explorer. So you can actually look up any gene and then see how it behaves in these two data sets in ALS cases and controls. And it will be available to accompany the publication. So we really hope that people will use it. So in conclusion, neuropeptide signaling was altered in these two data sets in ALS. Melanocortin receptor 4 significantly upregulated in both data sets would be a strong candidate target gene for future biomarker and screening studies. Our ALS Gene Expression Explorer provides access to gene level results to the wider community. Collaborators and funding slide. Heather is a PhD student who is first author on this publication. She has done a lot of work that I have presented here. And thank you to everybody else on this slide.